started. All right, thank you very much, everybody, for being here today. This is the second iteration of the extended bioinformatics workshops, and I have called on first Clement to talk a little bit about TE, but more importantly, right now we are here to hear Simona to talk a little bit about RNA sequencing and data analysis using R. So please, thank you all for being here, and thank you for the Vitali Lab. Dr. Winton's lab, for this opportunity and allowing you to be here to share your knowledge with us. So thank you very much, Simona, for being here. Join meeting club, Simona, welcome. Woo.
Okay, now you should hear me. Okay, as I was saying, from the link that I sent in the chat, you can access both the art demo that we are going to discuss and the wiki pages with all the material for the workshop. So I'm going to open the wiki page. Here you can find the home of the workshop. And this workshop, as Michele told, is going to be about differential expression analysis. So I'm going to give you a little bit of an overview of RNA-Seq technique. And the main aim of this workshop is to give you the materials and all the best practice in order to perform your own DEG analysis. So if you have your own data, you can follow along with your own data. If you don't have data, no worries. There's still, I have example data for the demo. What I want to disclosure before starting is that unluckily, this is not a workshop about statistics, neither an introduction or R. So if you're not that skilled in both statistics and R, if you have more questions about it, we can discuss at the end of the workshop. And this is neither a workshop, this workshop cannot cover all the RNA-Seq data collection. So we are going to start from the read count metrics, and then going to define the read count metrics in a little bit. So hopefully at the end of this workshop, you're going to understand the basic of RNA-Seq data analysis, and you will be able to know the basic steps to perform differential gene expression analysis. In particular, we are going to discuss 3 R packages that are called LIMA, DSeq2, and EDGE-R. The first 2 are going to be discussed also in the demo. The third one not, but there's still code available in the materials. And then what I really want you to make your own knowledge is how to perform the step-by-step analysis and how to manage your data and how to interpret and visualize your results in order to be aware of what you are doing with your data. So we are going to start with a little bit of introduction for the RNA-Seq technique. That is a high throughput sequencing technique that is able to capture, I could say, a picture of the gene expression by reading and quantifying the mRNA molecules that are present in a sample. So overall, is a very used technique, both mainly because it's high sensitive, so it can detect a very large range of expression levels, including the low abundance transcripts.
So you may not share.
Oh, I'm sorry. I'm going to share. Okay, now you should be able to see also my slide.
Yes.
Okay, nice. Okay, so another quality of this technique is that you can perform comparative analysis with the output, with the data output, and this is what we are going to discuss today. So overall, just keep in mind that the RNA-Seq technology is characterized by a sequence of actions. Both of them, all of them requires a lot of data quality control. And again, we are not going to discuss about that in detail, but here I link all the steps that involve sample preparation, sequencing, read alignment, and quantification of your data with all the links of the previous workshop that discuss these passages in a very detail. So another thing that I want to point out is that since we are going to start with the read counts metrics, you need to know if your data were collected with single cell RNA-Seq or bulk RNA-Seq. The main difference is that with bulk RNA-Seq, you capture the average gene expression across all of your cells that you analyze with the RNA-Seq. Whereas with the single cell RNA-Seq, you analyze the gene expression at the individual cell level. With this workshop, we are going to discuss about the bulk RNA-Seq. And so what actually is differential gene expression is a statistical approach that is used to identify genes whose expression may vary between conditions. Usually with a medical biological background, you want to compare, for example, disease expression level versus control or treated expression levels versus control. In the demo, we are going to discuss a group of treated people versus a group of control people. And here you can also you can also read, when you have time, some of the experimental design consideration if you are interested in the very first part of the discussion. So how to actually prepare your data, prepare your cells and everything in order to obtain high quality RNA-Seq data. Okay, so now we can actually start talking, going a little bit more into practical aspect of this analysis. So we are going to start by creating and uploading our read counts metrics. As I was saying, this is the starting point of our analysis, and the metrics has to contains all the genes expression for each of the samples that you have. And you need to be, you need to keep in mind that all your genes need to be in the row of your matrix, whereas all of your samples need to be the column of the matrix. What is also needed for this analysis is a metadata table that contains some of the characteristics of interest of your samples. Here we are going to work with human samples. So in our metadata table, we can have age information, chromosomal sex, and the condition of interest. So now I'm going also to open the demo in R. Okay. So for this specific demo, we are going to work with the data collected for this article that is titled RNA-Seq Transcriptome Profiling Identifies CRISP-ALD2 as a glucocorticoid responsive gene that modulates cytokinin function in airway smooth muscle cells. To make biology a little bit simple, what they did in this article and what we are going also to perform is comparing 4 samples that were treated with a glucocorticoids treatment that is called dexamethasone, and we're going to call it DX to simplify it. And these 4 people were gene expression data were compared to control. So we are going to import the data. The data were, you can easily import them by following this little box. And when you import the data, you see that, and you look at the dimension, you see that we have almost 40,000 gene for 8 samples. And when you look at the at how your data frame looks like, you see you have again the genes as the rows of your matrix and all the sample as the column of the matrix. Here, that are the that is the original data frame, we also have the very first column that is with the gene names. And as far as the metadata, when we upload them, we see that we have, again, 8 rows with 4 columns. And the first column is our ID for all the 8 samples. And then some information that is the first column, the one is of interest, describes our condition, that is control and treated. And then there is information about the cell type and the GeoID. We are going, for these analyses, we are going to look again at treated versus control, and then we are going to discuss a little bit about also the cell type. So what I want to stress with this tutorial, and I may be a little bit picky for that, is that you need to take care of your data very well before analyzing them, because with differential expression gene analysis, you almost always have results. But when you obtain results, if your data were not treated in a very in the right way, you can have like not biologically significant results. So first of all, once you have uploaded your data and you know that you have almost 40,000 genes for 8 samples, what you want to look at is, first of all, if there are missing data. So usually it really depends on how you obtain your data. So if you were the one that made the experiment and did the protocol to collect the data, you already know how, all this specification of the protocols. If you take data from a company, usually they provide documentation and with all the information that you need in order to know how they handle missing data. So in case, usually if they're like, if they're characterized by non-values, you can just count how many non-values you have. For this specific example, there's 0 missing values in our read count table. But let's suppose we have some missing data. What you can do is replacing them with 0 if the hypothesis that you made is that the non-values means that that gene has no expression. So just going a little bit here in how to handling missing data, there's a box with some tips on what is better to do and what is not recommended. And overall, even if maybe you are a data science and you really like imputing your data, this is not really recommended for RNA-Seq. Mainly because the missingness can be due to many reasons, like technical failures or bioinformatics processing errors, or again, low expression levels. So since when you don't know the reason why your data are missing, it is better not to impute them. And again, you can replace them with 0 if you know that missing values means no expression, or if you feel confident enough to suppose it. You can always filter genes with high proportion of missing values. In our demo, we are filtering our missing data that has more than 20% of missingness. So we are getting rid of all of the genes that has more of 20% of missingness. And then keep also in mind that as is specified at the very beginning of the demo, we need to take note of all the steps that we are doing. So also, when we are getting rid of the missing data, we should still take in consideration of writing down all the genes that we are getting rid of. And
No question. Is 20% the default value that you will take?
Yeah, it's usually the default value. I will say that it also depend on the amount of data that you have. If you have a smaller data set, maybe 20% is maybe you want a smaller threshold. So,
and on this question, do you look at the balance when you think, if, for example, you have a gene that's only expressed in one half of your data, for example, and not the control, like in the treatment control, do you want a little bit of expression? Like, would you keep, let's say, all the control are 0 and all the treatment are, would you keep this gene?
I would say no, because especially if it is a situation like that, I would say that data are not missing at random. So there is a technical issues due to that missingness. And also if there, for example, if you have only the expression for the treated and you don't have the expression for the control, it should not, there is like no way you can do the comparison. So you need to get rid of that gene. But yeah, no, that's a good question. I think that overall, again, the threshold, the recommended threshold is 20%. But if you're working with a smaller data set, when I say smaller, I mean based on the number of the total number of genes that you have. So if you have like fewer genes, of course, you need to decrease that threshold. And another thing that can happen is that for the genes that maybe have like low expression or they were not detected by the machine, is that you have all 0 expression for all of your samples. That is the case of this data set. So when I did check about how many genes has expression equal to 0 for all the samples, I found out there were almost 40,000 of genes with all 0 expression that are not giving information about the differences between treated and controlled. So I removed all of these genes, and overall the final the read count matrix has 25,000 genes for, again, 8 samples. Okay, and then before going into the clue of the analysis, there's something I want to say about how to visualize your data before analyzing them. Because what we are looking for after the missingness of the data is how to handle outliers, if there's any. So what is recommended to do, and I'm going to, to keep up with the theory on the left, is what is recommended to do when you want to visualize your data is to log transform your data, because as you can see, if you plot the expression level for each of the 8 samples that you have, you are not going to appreciate the expression level. Whereas if you log transform in base 2, you skew the data and you are more able to make comparison between the samples. And there are a lot of data transformation that depends on the number of samples that you have and the R packages that you want to use for your analysis. And we are going to discuss about this in a little bit. Another aspect that you need to consider when before analyzing your data is to check for variance homogeneity. That means that you usually low count genes can carry a lot of variance. So you need to understand the reason of your variance. So what you can do, and since we have only 8 samples, this is something that can be done iteratively. I did only plot the comparison between 2 samples, but what you can do is do a pairwise scatter plot, and in order to inspect the count similarity across these 2 samples. And as you can see, the more aligned the data are, the better the quality of the data is. Here you can see there's a lot of genes that has a 0 expression for one of the 2 samples, but that does not have for the others, and the same. And then another thing that is recommended to do based on how you want to model your data is to look if there is some dependency between the mean SD of your data and the variance. So what you want to do is just to plot the mean expression of all of your gene on the x-axis, and then their standard deviation on the epsilon axis. And what you can do, what usually people do is just plot a subsample in order to look if this subsample follow the distribution of the data. Here I plotted all of them in order to see how they looks like. And what is good is that at a low mean level, there's not so much variance in the data. There's a little bit of variance when you increase your mean, but still overall, the data are kind of following the distribution. Okay, and then there's other check that you can do, and everything is reported in the materials, especially for the data that you want to analyze with this DSEQ2, especially because you are transforming the data in a different way, specifically to that model. Something that you can do, the last thing that you can do before analyzing the data is to look for pattern in your data. And we are going to discuss pairwise correlation, hierarchical clustering, and PCA, that is principal component analysis, to have the last overview of our data. And again, this is recommended to do in order to look at the variability of the samples, and again, look for outliers in your sample. So for the pairwise sample correlation, here is pretty easy to plot because we have only 8 samples. So we are looking at sample to sample variability. And specifically with this plot, we are looking at how similar the sample are in terms of gene expression. You can, since it's a mirrored matrix, you can look only at the half part of it, and you can see that the samples are the gene expression of the samples are pretty similar. And this is good because if you have very dissimilar samples, that can be a sign of some technical issues with the data collection, with the collection of your data. So you want your gene expression overall to be more similar, but not identical. Otherwise, you're not going to detect any differential expressed gene. Even if you can still appreciate it here in the pairwise sample correlation. I reported the hierarchical clustering plot. And with the hierarchical clustering, you are look if there are some clusters in your data. Ideally, you want your treated samples to cluster together, and you want your control sample to cluster together. But it's not always like this. Here we can see that, and with hierarchical clustering, you can decide where to cut horizontally the plot in order to count for clusters. And this is the same information that you can appreciate in the pairwise sample correlation tool. And lastly, what is actually my favorite is the principal component analysis. That is just a dimensionality reduction technique that helps you to summarize the variation in your data set in fewer components. in particular, you can see how much variation each component is able to explain. And here I reported as a plot where you can say that where you can see that the fourth principal component is able to explain almost 40% of the variation of the dataset, and that's good. Whereas the second is explaining the 15% of your data. If you have more interest in your principal component, you can then access each component and see how which variables are characterized that components. Because again, it's a dimensionality reduction technique and basically works by doing a linear combination of your variable for each of the component. So you can access component one, a principal component one, and see how your genes take part into that component. And then what you can do, of course, is plot your data in the new space of PCA. And here I plotted the 8 samples gene expression divided by control, that are the pink one, and treated, that are the light blue one. And there's separation between control and treated. That is good. That means that the condition is able to explain part of the variation of the data. So I'm expecting to have at some of to find differential expression gene among between these two groups. You can also plot your new your data in the new space divided by cell type. So here I reported condition and cell type together, and you can see that again the main variation is carried by the condition and not the cell type. Okay, so I think that with this we are almost ready to actually perform the G analysis. To summarize our pattern research, overall, the pairwise correlation is used to reveal sample similarity at a higher level, whereas hierarchical clustering wants to group similar sample, and you can use it to identify outliers, because if you have like a cluster with only one sample, maybe you can look better at the gene expression of that sample to better understand the reason why that sample is clustering alone. And then there's the PCA that reduces the dimensionality and summarize the main sources of variation of your variables. And what I also would add with the PCA is that it can help you to find outliers too, because when you plot your sample in the new space, if you see that one of the sample is very far away from all of your other samples, that may be due to some outliers in the gene expression of that sample. So far, do you have any question about how to handle the data before starting the differential gene expression analysis? Okay, so I guess we can. Do
you have to generate it?
There, you mean for the demo or in general? Oh, for. Yeah, so the metadata are not related to the output of the RNA-Seq. So usually what happens, for example, if you are doing some experiment with mice, you have a table with all the information about your mice, such as month, sex, and other that you are interested, and then you have the gene, the read counts data apart. So yeah, they're always separated. Okay. So for this, as I said before, for this demo, we are going to look at the lemma and these Seq2 packages, and we are not going to discuss EdgeR, only because EdgeR perform very well with larger data set, and we have only 8 samples for this demo. So it is better not to analyze the data with EdgeR, even if, again, if you have your own data, you can, there is a section, and you can look at the code if you're interested in look at data preparation for EdgeR. Overall, what you need to know about DG analysis is how to transform your data based on the tools that you are using. And in particular, even if I told you about the log2 transformation for data visualization, you don't have to perform it with lemma and Seq2 because they have their own data transformation normalization. The data transformation I told before, I discussed before, is mainly for data visualization and is mainly for better represent the variation in your data and better visualize your data. So for the DG analysis, both in lemma and the Seq2, we are going to start with data that are neither normalized or transformed. In particular, for lemma package, you need to convert your data, your row counts into log2 and then count per millions. Whereas with the Seq2, you need only to perform size factor normalization before analyzing the data. So in the demo, we are going to discuss first the analysis in lemma. So to do that, again, we need to take our read count metrics, and then we need to define which is the condition that we want to compare in our analysis. And to do that, I'm going to discuss to better discuss how to build the model for your analysis. So to do that, always on the left part of my screen, again, here you see everything that you need to prepare your data for the analysis, and then other recommendation about how to normalize your data and all the type of data normalization. And then in the third chapter, there's the actual differential expression analysis. And now that we understood how to manage the data and what we need to look at for account for the quality of our data, I want you to know that the differential gene expression analysis, what you mainly do with both lemma and the Seq2 is do linear regression analysis for each of the genes in your data set in order to see if there is statistically a significant difference between the treated and the control expression in the expression of each of the gene. So again, each of these methods use regression modeling. And then what is up to you is how to choose your model for your regression, and in order to take into consideration also the variability of your data. The output of this analysis is a list of p-values that tells you which of the comparison is significant from a statistical point of view. And so for the experimental design and how to build the model to put into the lemma or the Seq2 tools, you need to take into consideration what you want to model in your regression. That means that usually with linear regression, you have for each of the gene, you have the expression of that genes, and you want to regress how the condition that for us is treated versus control impact the gene expression. But in your model, you can add other information if you know that there are other variables that carries variation in your data. So usually you can build a model only using the condition, and you are looking only at the effect of your condition. But sometimes, again, you usually find the information on the documentation provided with your data or on the protocols that you applied your on your experiment. You usually also take into consideration also the batch effect that is usually carrying a lot of variation. And then there's there may be other known factors that you want to include in your model. And it's very important, if you know all of the origin of the variation of your data, it is very important to include them into your model, because you don't want your results to be confounded by some of the variables that you may not include in your model. So usually you also consider age, if you have age, or you may consider sex based on the hypothesis, what you want to test with the differential gene expression.
In the model for DSEC2 or HR, or do they have their own ways of defining the model?
So it's almost the same. The only difference is that in Lemma, you have a variable that is your model variable. So you create your model variable, and then you perform the linear regression saying, use this model that consider these variables. Whereas in DSEC2, you have everything together. So you're performing the analysis, defining the model in the same function that you perform the analysis.
Thank you.
Sure. So, and so once you know everything that can impact the results of your analysis, you can consider all of these variables in your model. Something that I found interesting is that there are also some regression models that are called interaction models, in which you can take into consideration about some of the interaction of your variable. So if you know that maybe there's interaction between a genotype and the treatment effect that you are looking at, you can model that in your model, just adding a multiplication variable. And then something that also you need to keep in mind before performing your analysis is whether you have paired samples in your data set. Paired samples are usually obtained with paired RNA-Seq. That means that for each of the sample in your data set, you have two gene expression, one that analyze and map all of your gene starting from the very beginning of your genomics to the end, the other that do the opposite. So it starts from the end of your genome and goes to the very beginning. That means that, again, for each of your samples, you have two columns. And so in order to take into consideration the paired samples in your analysis, you just need to add a variable in your model that is called subject, but can be also called sample. But again, you add it in order to take into consideration the fact that a sample can have more than one gene expression in your data set. So you need to take in consideration the subject also in your model. Something that you can do also with this kind of a regression model is to take in consideration multiple comparison across groups. So as I told you, in our data set, we are comparing treated versus control, but it can happen that you want to compare more than two groups. And so you want to compare maybe like control, treated, and people at risk. And so you have three labels for your data. To do we're not covering the multiple comparison analysis with this demo, but I did link two links for to the documentation of both of the packages, and you can have a look there and see how you can modify your model in order to take into consideration multiple comparison. But basically, if you are more familiar with statistical analysis, with statistic and statistical tests, what you need to do is just change your statistical test because you are considering more than two groups. And so overall, what is unique to each of these packages is the how to use them based on the sample size of your data set, how they model the distribution of your data, which kind of statistical tests they're using, and which kind of transformation of the data they're applying. So for DSEQ2, again, this is recommended for smaller sample size, usually lower than 12, and they model they suppose that your data are distributed with a negative binomial distribution, and they do the wild test. And for the data transformation, they apply the sample-wide size factor for each of the gene expression. Whereas in Lima package with also the VUM package, you can kind of apply this pipeline no matter your size of the data set. And they do want the log normal distribution of the data, and they apply the eBayes moderation with a different transformation of the data, because you need to transform the data you need to apply a log2 transformation counts per million for your data. And you also need to take into consideration the precision weights associated at each of your samples. And lastly, EdgeR, as I anticipated earlier, is good for a larger sample size with, again, a negative binomial distribution of your data. And here you find more information about the testing approach and transformation. So going into the analysis results, first of all, we are going to look at the Lima output. So as I told you, here, you need to specify which model you want. That means that for each of the gene, that your gene, I'm going to let you see the model again, your gene expression will be the output variable. For each of your gene, you are going to regress out. You need to specify which variable you are going to regress out. And for these, we are going only to model our group comparison that, again, is treated versus control. And here is the design model that we need to apply to the actual analysis. And then what is also recommended, but we already done it, this is redundant, but we want to filter out lowly expressed genes. But this is done taking into consideration the fact that we have this specific design, and so we want to compare treated versus control. And then you transform the data using the VUM function that log transform the count per million and estimate mean variance relationship in the data. And this is how the data looks like after the data transformation for Lima. Again, genes in the rows and samples in the columns. And lastly, what you need to do is fit the model. So you specify the design, and you give as an input the VUM object and then you apply your statistical test, and you can visualize the results. So the results usually looks like this. So a very long list that contains each of the genes for your rows with a lot of scores. The main interesting one is, of course, the log fold change score that tells you that quantify how different the expression in the treated group is versus the expression in the control group, and is a log transformation. Here on the left, I reported also the mathematics under the log fold change. That is pretty simple. So it's the log transformation of the expression condition, so the treated expression versus our control expression for each of the gene. A higher log fold change, a positive one, means that that gene is upregulated in the treated versus the control group, whereas a negative score means that that gene is downregulated in the treated group versus the control group. So the up and down regulation is linked to the expression number of each of the gene. What also we need to take into consideration is the p-value that tells you if that difference is actually statistically demonstrated. So you have the p-value, but since we are looking at almost 25,000 gene expression, what you want to do is to adjust your p-value for all the comparison that you are doing. And so what I do recommend is to use the adjusted p-value. This is adjusted for the false discovery rate. And so what you want to do with this output is to filter for all the genes that have an adjusted p-value lower usually than 0.05. That is the threshold that is usually used. And then look at their log fold change to see if that gene is up or down regulated in the treated group. There are other information that comes with this analysis. That is the average expression. That is the second column of the output that just tells you the average expression of the gene across all the samples in the analysis, no matter if they're treated or control. The T, that is a variable that is used for the statistical test. And then the B variable, that is the log odds of differential expression, that is called the B statistics. And basically it estimates the likelihood that that gene is truly differentially expressed based on the model that we use. That our is like the simplest one because only consider the condition as a variable. And how to interpret that is that the higher, the better. So you want for sure an adjusted p-value lower than 0.05, but you want also a high b value. And for the log fold change, usually you look for upregulated genes that are defined with a log fold change greater than 1, and downregulated genes that are defined with a log fold change lower than -1. So I will go very quickly with the DESeq2 analysis because it's pretty similar. The only thing is that before analyzing your data, you are not going to create a VUM object as earlier, but we are going to create a DESeq2 object, just giving the row counts data, and again the design that you want for your data. And then you're going to apply the normalization and inspect also the normalized counts in order to see if you have high variance in your data or not. And then when you want to, again, as you can see, the way that DESeq DSeq to transform the data is very different from the output of the VUM object. And then when you want to apply the DSeq2 analysis, you just run this pipeline with the DSeq function that wants as an input a DSeq object, and then you can you can visualize your results. Here you can have here we have a little different results, variable as results. In particular, DSeq2 is able to count how many genes are upregulated in the are significantly upregulated in the treated group, and in this case, we have almost 6% of the total genes that are significantly upregulated. You have the same measurement for the downregulated genes. We have almost 5% of the total genes that are significantly downregulated. Here is also DSeq2 is also telling us that it detects some of outliers, even if we were not able to detect them with our visualization technique, that are less than 1% of the total. And then just low counts genes, that means that overall there is like low expression for the genes. And when you visualize the results, they're pretty similar. The output is pretty similar to the lemma one. You have the base mean variable that is again the average of the normalized count values. In this case, divided also by the size factor of all the samples for each of the genes. And then again, we have our log fold change that tells us which gene is up or down regulated. And then there's this variable that is called LFC-SE that take also into consideration, take also the standard error into consideration. And so you can is a quality check score that you can have for your statistical analysis. Again, the stat that is used from the statistical tests in order to apply the test. And again, we have the p-value and the p-value adjusted. So once we have performed this analysis, again, our results are it's a very big table with a lot of rows, one row for each of the gene that tell us which the gene is dysregulated and if it is up or down regulated. And again, if this dysregulation is significant in our data. And so what is recommended to do is to apply at least 2 of these tools and then look at how many genes are shared between these 2 different results. And so to be more clear, what you can do is just to plot how many genes you detect with DSeq2 and how many genes you detect with lemma, and look at which one are shared among the 2 approaches and which are not. And here, as we can see, we almost find 2,000 significantly dysregulated genes with DSeq2 and almost 1,200 genes with lemma. And lemma looks like a subset. The genes detected by lemma are almost a subset of the genes detected by DSeq2. Another way to visualize it that I personally really like, especially if you want to compare more than 3 groups, is to use the upset plot. Because one of the limitations of this plot that is called Venn diagram is that when you have more than 3 groups to intersect, it became very messy, and you're not either like able to understand all the interaction among the groups. So an easier way to do it is just to use the upset plot that tells you with the dots which group you are considering. And in this case, we are considering the lemma the genes detected by Lima and the gene detected by DSIG2, and you can see how many are in common and how many are specific to DSIG2 and specific to Lima. And so again, what you need to keep in mind when you want to apply your own pipeline is how you want to transform your data, how many samples you have in your data, and which tests you want to apply, and of course, which model you want to use to design your experiment. And lastly, here I reported also more tools for DEG analysis, especially if you don't like to code. There's some of them that you just give them your table, your read counts table as an input and your metadata, and they perform the analysis for you. And lastly, and very quickly, we are going to briefly discuss about how to interpret our results, even if we already done it from a statistical point of view. Now we are going to discuss how to show your results to your PIs or whoever you want to show your results. So something that is very common is called Vulcanoplot, and it plots the log2 fold change versus the negative logarithm of the p-value in basis 10. And how you interpret it is based on the p-value threshold that you select. You divide your data horizontally, and everything that is upper the threshold that the p-value threshold that you find is statistically different. But then you need also to consider the log2 fold change, because as I told you, for upregulation, you take into consideration a log fold change that is greater than 1, and for downregulation, a log fold change that is lower than -1. And so you are also vertically divided your data based on the based on your log fold change. And here, as you can see, the gray one are the non-statistically significant genes, whereas everything that is greater than 1 log fold change and upper to your p-value threshold is in red, and these upregulated genes, whereas all the blue dots that you see are the downregulated genes. So very quickly, another way you can, this is like the main, the most representative plot of your data, the volcano plot that makes you also, gives you a qualitative idea of how many genes are upregulated and how many genes are downregulated. What is also interesting to look at is what is called MA plot of differential expression gene, in which you are plotting the average expression versus the log fold change. So here you can see which genes are upregulated and how big their expression is. And again, the red dots that you see here are the significant upregulated one. And you can see that overall, both for the upregulated and downregulated in blue, their expression is the main genes expression is before 5,000, and this is of course count per million. And this is also another way to do outliers detection after your analysis, and is another way to do quality control of your results. And if you see that are, for example, also these, there are like dots that are very far away and a very average expression, a very big average expression. Maybe you want to get rid of that genes, or maybe you need like to go back and look at how you manage your data and better detect outliers in your data. And lastly, what is easy to do when you have few samples is just to plot your dysregulated genes in a heat map, where the red again means upregulation and the blue means downregulation. And you can plot also other information. Here you have again the genes in the rows, the samples in the columns, but we also have the information about the groups. And the main limitation of this plot is that if you have a bigger sample data set, it can be very difficult, first of all, to plot all the columns, so all of your sample, but also to plot all of your genes. Here, for example, I plotted only the first 40 more dysregulated genes. And so what also is recommended when you have a very large data set, so many samples, is just to plot at their average expression. So from a qualitative point of view, you can see the main dysregulation in the control and in the treated group. And so you can see in the control that most of the dysregulated genes are downregulated compared to the treated group. So that these here overall, what we can see is that in the treated group, there is an overall upregulation of the dysregulated genes. And here there are other very useful links to see other plots. And with this one, with this plot, I think that I'm officially done with a little bit of late. But if you have any question, just let me know. If also reach out to my email address. And don't worry if you haven't seen any fancy application of these analyses, because in the next workshop next week, we're going to discuss some of the nice application that you can do with this information. And that's it. Yes, we are going to see KEGs both in R and also some tools that perform KEG analysis just without coding. Okay, thank you.